Welcome to our first Year 10 science video on genetics, and this video is all about genetic coding. Okay, first up, as we're looking at this whole idea of this genetic coding and these messages being sent literally through genetics and information transferred through genetics, we are going to look at DNA. I'm sure this is something that you have all heard of, but what is it? Where is it found and what does it do? So first up, DNA. Where is it? Okay, well, cells are the building blocks of all life forms, and you've got a 3D diagram of a cell there, which you've probably seen before. Now, they contain many structures that contain the actions of the cell. These functions are all coordinated and controlled by the nucleus, and it is in the nucleus of our cells where the DNA is found. Okay, so it's found in the nucleus of cells, but what on earth is DNA? Well, the term DNA, or the acronym DNA, stands for deoxyribonucleic acid. Don't forget that one. Now, DNA contains the instructions for the cell and for the entire organism that it's found within. It contains the code or instructions about how that individual will look, how it will behave, and how it will function in the world. DNA is usually a very, very thin, long, string-like structure that is almost invisible. We call the shape of the DNA a double helix, and you've probably heard this and seen diagrams as that such on the slide. You know, it's often referred to as like a twisted ladder, that double helix. The structure of our DNA. Why does it have that double helix? Why does it look like that twisted ladder? Well, our DNA molecules are made up of what we call nucleotides. Okay, and this is like one of the rungs on that ladder, and this is one nucleotide in red there. Nucleotides have three parts. Okay, they have the nitrogenous base. Okay, so you'll see the base here, and this is the base here. It's a G because it's a certain type of base in this diagram. Okay, but that is the base group. It's then got the deoxyribose sugar. Okay, so here's our sugar, and here's our sugar there on the chain. And then it's got the phosphate group here. So there's the phosphate, okay, and we quite often call this the phosphate backbone. So the sugar and the phosphate make up the sides of the ladder, okay, also known as the sugar-phosphate backbone. So the sugar-phosphate backbone of the DNA. The bases of the two strands kind of join together. So you've got base 1 here, you've got base 2, and they join together. So this is one nucleotide over here, this is a nucleotide here, and they're joined together through these bases. Okay, and they make like the rungs of the ladders, so the steps of those ladders. The pairing of these bases is what we call base pairing, and that's something we will look at a later date. So nucleotides, they're arranged in such a way that they make that double helix, that twisted rope ladder as such. Okay, and here we've got a diagram of that twisted rope ladder, and you can see those different colours along it being those different bases that pair together. Okay, and these are actually the names of the bases. Now the uprights of the ladder, as we said, are made of those phosphate sugar groups. Okay, and here we've got it, phosphate sugar, the phosphate and the sugar of that first nucleotide, the phosphate and the sugar of the second nucleotide, the phosphate and the sugar of the third nucleotide. The rungs are formed by the base pairing. So I'm just saying the same thing, but just showing you different graphics so that it kind of makes sense. So the base pairs here being those rungs of the ladder, or the steps of the ladder. Okay. All right. Still looking at the shape and structure of our DNA, but here we're actually looking a bit further at those bases and getting a bit of an idea around this base pairing. So those rungs of the ladder, the steps of the ladder, are two of these bases pairing up together. Okay, now there's only 4 different bases in DNA, so no matter what's happening along here, there's only 4 different bases, okay? And that's adenine, guanine, cytosine, and thymine. Now, still looking at this structure and shape. The bases in the DNA, adenine, guanine, cytosine, and thymine, are referred to as complementary. Okay, adenine will only ever bond with thymine, so A to T. Okay, A to T, T to A. So they're complementary to each other. They will only ever bond together. Guanine will only ever bond with cytosine. So cytosine, guanine, okay, guanine, cytosine. So they will only ever bond together. This is known as our base pair rule. Alright, so now we've looked at DNA and its structure and why it looks like that twisted ladder as such. Let's move on to looking at chromosomes. Okay, you can see our double helix here, and you can see it twisting around these proteins that are called histones to make these coils, and these coils then coil into what we call supercoils, which are all part of our chromosome. So basically we're saying our chromosomes are made up of DNA. Okay, so as you recall, DNA is usually very thin, long, string-like structure that's almost invisible. However, when the cell needs to divide, the DNA kind of winds itself up. It's supercoils, okay? So that's what we're talking about here, this supercoil. Supercoiled DNA takes the form then of chromosomes, which we can see, which is then visible under a microscope. This helps the DNA to stay organised whilst the cell's dividing. Now we'll have much more of that when we move on to part 2. Okay, but during cell division, the last thing you want is a heap of DNA that's all mixed together by spaghetti. We need it much more organised. So just before cell division, our DNA supercoils into chromosomes ready to divide. And those chromosomes can be seen under electron microscopes. Alrighty, so in this graphic here, we can see our cell, and it's identifying that in our cell we've got our nucleus, and inside our nucleus we can see that we've got our chromosomes, and we're saying, well, those chromosomes are made from DNA.
We'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. Next, the nucleosomes are packaged into a thread. The end result is a fiber known as chromatin. This fiber is then looped and coiled yet again. Leading finally to the familiar shapes known as chromosomes, which can be seen in the nucleus of dividing cells. Chromosomes are not always present. They form around the time cells divide when the 2 copies of the cell's DNA need to be separated.
Okay, so when we start looking at chromosomes, let's really think about what they are. So we've already said that chromosomes are made from DNA, so constructed from that DNA. Okay, so DNA can be compacted into these thread-like structures that are called chromosomes, and you can see that here on our diagram. Okay, so our chromosomes are important because they contain the genetic code for the organism that those specific chromosomes are found in. So the chromosomes within your cells code all of your genetic information. The chromosomes found within the cells of a frog code the genetic information for that frog. The chromosomes found in the cells of a potato plant code for that potato plant. Now, in the human body, each cell contains 46 pairs of chromosomes. 46 chromosomes in 23 pairs. If they've got 46 pairs of chromosomes, it's a completely different organism, it's not human. Okay, so we've got 46 chromosomes paired up into 23 pairs. The only exception to this are the sperm and egg cells, so the gametes, and that's because they contain 23 chromosomes each, so that during fertilization, that fertilized cell then contains 46 chromosomes because the sperm and the egg have joined together. And the other exception to the rule is our red blood cells, and that is because red blood cells do not contain a nucleus, so that they can carry more oxygen around in their hemoglobin. A nucleus would be wasting space in a blood cell. Here we've got some pictures of different representations of chromosomes. Okay, some are electron micrograph pictures, others are computer generated of how chromosomes look. Okay, so we've said that DNA coils up into supercoils and forms what we call chromosomes. So where do genes come into this? What are genes? Genes are sections of DNA that code for specific proteins. Okay, so effectively, genes are sections of our chromosomes. Now, each chromosome can have over 1,000 different genes on them. Each gene code contains different information. Now, it's for specific proteins, okay? And proteins contain many of the characteristics and functions within the body. Proteins include the structural materials that build up your cells and tissues, most hormones, and all enzymes. So enzymes are produced from proteins. All right, genes and chromosomes. What is the relationship here? Now, the chromosomes contain specific instructions for all different characteristics and traits. For example, eye colour, okay, or natural hair colour, or protein functions, so such as making those enzymes. This code is divided into different sections along your chromosome, which are called genes. Remember, there are 46 chromosomes in almost all human cells. We've already spoke about the exceptions to this. So there's 46 different chromosomes in almost all human cells. Each of these chromosomes contain many, many genes. Alrighty, so at this point, can you explain the relationship between our DNA, our chromosomes, and our genes? Think about it. Can you explain that relationship? Each chromosome has a partner that is the same size and shape. Now, this is apart from our male sex chromosomes. These are called homologous pairs. In human cells, there are 46 chromosomes, or 23 pairs. Okay, so just repeating this to just remind you. Homologous pairs contain the same genes as each other. So each pair contains the same genes. One of the pair came from the maternal cell, so the egg cell, and one of the pair came from the paternal cell. Okay, so we call them the maternal and the paternal chromosomes. They're not identical, as the sequence of bases on each could be different, but they are a pair. So how can they be different? Okay, and if we look at the diagram here, we've got two chromosomes. We know that one is the paternal, one is the maternal. So one's come from the mother, one's come from the father. And this occurs during fertilization when the sperm and egg combine. Now, so how does this make the difference? And you can see in this pair here, we've actually got an allele, so a gene to a certain extent for blue eyes, and we've got one for brown eyes. So the gene appears at the same point on each chromosome, but they're different. Okay, so the trait is located at the same point on each homologule. Okay, we've got a chromosome pair and the same gene trait at the same point. Okay, but the different forms of the specific genes are what we call alleles, and we're going to have more about this in section 3. Okay, now some important terms here. You'll see these terms haploid and diploid. Okay, so the number of chromosomes in body cells is the diploid number. Okay, so this means there are 2 sets, diploid. In the gametes, there are half the number of the chromosomes. Okay, so there's 23 chromosomes in a human gamete. Okay, and we call this the haploid number, the half number. Okay, so diploid, D2. Okay, so that would be a full set of chromosomes there. Okay, here in our gamete, we've got half the number, so that haploid number. Now the haploid number is quite often called N, and the haploid number is called N, and the diploid number is 2N. Okay, so N, 1 set, 2N, 2 sets. Of the 46 chromosomes in a human cell, 2 are sex chromosomes, the ones that determine whether the offspring will be male or female. The other 44 chromosomes are not sex chromosomes, and they're what we call autosomes. In human females, the sex chromosomes are a pair of X chromosomes, so XX. And you can see on our diagram here, we've got a female XX. And we call that a homologous pair. They are the same. In males, the sex chromosomes are an X and a Y. Okay, so we've looked at chromosomes, we've been looking at types of chromosomes. We're now going to look at what we call karyotype. Okay, and karyotype is all the chromosomes arranged in their homologous pairs. So here we've got them all in their homologous pairs. And in a slide earlier, we said that the pairs all generally look very similar in length, except for the sex chromosomes. And we've got them down here, and you can see that that X chromosome is much bigger than the Y chromosome. They are numbered by their size from their largest down to the smallest. Karyotypes make it easy to identify genetic disorders and the gender of an individual. So if you align the chromosomes into the order of size, you can then locate different genes along the way, especially if you're looking at genetic disorders or the sex of the offspring. So is this individual we're looking at here male or female? So we need to look at this pair here, and that pair is an X and a Y. So if we look at the karyotype though, how many homologous pairs have we got? We have got 22 pairs, because that is not a pair at the end. What is our diploid number? So what is 2N? Okay. Alrighty, so as we finish this unit, these are some key terms that we've looked at in this video. hope you enjoyed watching. Thank you, and our next video will be done very soon.